The project name is Innovative Cancer Detection Mechanism. In this method, we are demonstrating an effective and useful method for detection and the removal of cancer cells from the blood. Cancer cells are fast growing cells. Once the tumor is formed, after some time the tumor tends to pass into the bloodstream and it will spread to the various organs of the human body. Now in this method we are going to remove these cancer cells by designing a specific antibody for the cancer. Now the technique which is used here is flow cytometry. Flow cytometry is used to analyze the blood. Now in this technique, normally the technique is similar to the hemodialysis. In hemodialysis, the blood is collected from the vein through a pump onto the dialyzer, where the waste material is removed from the blood and the blood is purified and sent back to the human body. Now the waste material here is the cancer cells or circulating tumor cells. Now we have to develop a technique of detecting and removing the tumor cells. Now in this, see, we collect blood from this vein, from the hand, and instead of going to the dialyzer, that pipe will come into this a funnel kind of arrangement. The funnel neck is arranged in such a way that it is very narrow, that only one cell of the blood content can pass through it at a time. So when it is passing through it, this is focused, this is being subjected to a laser light. The laser is focused from here to onto the cell which is coming out of this thin funnel. So like there are three lasers: red laser, green laser and blue laser. Using this RGB, when we detect the scattered light, this is the scattered light detector. This is called a side scatter detector and this is called a forward scatter detector, since it is in front of this. So we have got a combination of forward scatter detector as well as the side scatter detector. Using the side scatter detector is given to analog to digital converter, and that is plotted onto the computer. When you plot on the computer, we will know, like, whenever something is falling from this narrow channel, the light beam is interrupted between the laser source and the detector, so we can count how many cells have passed through it. Then based on the characteristics or the reflection of this light, because each cell is of specific color, so we get information about that specific color from this scatter detector. So that is how we identify what are the normal cells. Like for example, the blood content is red blood cells, white blood cells, platelets, and in our case the tumor cells as well. Now the tumor cells will have a different morphology or the shape and size, so they will be detected based on this ADC values. These values are recorded onto the computer and a histogram is taken out of that. Based on this histogram, we can identify what type of tumor cell is that. Then based on that, we can get an devise an antibody, and that antibody is connected through this channel. Now this is an antibody. We are demonstrating the antibody with magnets and some metallic content of the ball. Like for example, in this we are demonstrating that the green balls represent the circulating tumor cells, whereas the other balls represent the red blood cells and white blood cells. Computer. Now the computer cable is to be connected to this Arduino board. You have to insert like this, then after inserting the connector into this, the board starts glowing. Now we will go to the monitor. We open Arduino IDE. Once Arduino IDE is opened, we will get the data. Now Arduino IDE is open. Now go to the serial monitor, which is on the right corner. Click on the serial monitor. You will start getting the data there, like that 805 and 764. This represents first sensor and the second sensor. First sensor is around 800, second sensor is around 750, like that. So now these values can be plotted. We will close the serial monitor and we will go to the tools, then we will go to the serial plotter. This is the serial plotter option which we have to select. Once the serial plotter is selected, we will get two waveforms. One is represented in blue, other is represented in red. Now for example, Now for example, if there is any object, now whenever the cell is falling from this, we will observe a small change in the output of this leaf. So we have observed that there is a small spike in there. So this spike indicates that the laser beam has been interrupted. Now I have interrupted the laser beam. Now you can observe there is a spike. So whenever there are objects are falling, so you will get these spikes. These spikes indicate that the number of objects that are falling. For example, if I am doing it slowly, you will know it. You will realize clearly. Now we are leaving a cancer cell into that, and we will observe the waveform here. Slow. Okay. Now you can observe there is a spike. The spike indicates that the laser has been interrupted. Now we will use other lasers as well. Now we will see the scattered light. ఈ పక్క పెడితే ఇటు అటు స్కాటర్ అవుతుంది లేజర్ మీద. Okay. Whenever the light is being scattered, you will get a different waveform. The waveform will be different for scattered light. This is for a red, green. Green. Now you place a red one. This is the default condition where the waveform is there. Now we are placing a red object, red color. For example, say red blood cell. We will get a different waveform. Now the scatter light is different. You have to observe that this is the normal pattern. After placing this, we will get a different pattern. Now you place another color. The next color is, what is the color?
Fluorescent.
Fluorescent. For fluorescent, you will get a different waveform. Based on the reflection of that object, we will get a different waveform. Like this, we will get data about these various objects. This is how we get information about the size and shape of the object. Now we will demonstrate. Once it is detected, it is passed through a narrow channel which is called a filter. It's called a membrane filter, wherein antibodies are coated with the membrane filter. The membrane filter will attract the cancer cells and will allow other cells to flow through. Now we are going to make these cells flow through this filter mechanism. Now let us see how it will flow through. Allow it to flow. You stir it and you do it. Do it. Parledan. Okay. Now we can observe all the blood cells, white cells and platelets have come to the other end. Whereas the green cells, which are cancer cells, they get attached to the antibody. Now this antibody will slowly remove that and it will kill those cells. Like this, we will continuously filter the cancer cells from the body, and after some time the cancer will be totally removed from the human body.
Primary colors.
We are using 3 types of lasers: red color, green color and blue color. These are called RGB or primary colors. There are several color schemes available, CMYK, RGB, like that. So but RGB is most famous. It is the simplest color scheme wherein any combination, millions of colors can be derived from the combination of red, green and blue. So one color will be more, other color will be less. Like that, by giving different proportions of the light, we will get all the colors available in the rainbow. Okay, so that is the reason we are using it. But whatever be the color of the cell, so that color will scatter. For example, if it is a purple will scatter say X amount of light, sideways as well as forward scatter. Another color will scatter another Y amount of light. So based on this, we will know the color of the object, the size of the object, the other features of the object. For example, as we know, like the cancer cells will have a different shape. Okay.